All right, Ninja Nerds, in this video we're going to talk about the adrenal gland. If you guys haven't already seen it, go into our endocrinology playlist and watch all of the videos up until this point. First one was on the zona glomerulosa, second one was on the zona fasciculata, third one was on the zona reticularis. In this video we're going to specifically be focusing in on the adrenal medulla, okay? So adrenal medulla is this inner part, right? And it's actually made up of neural tissue, because if you see, we're zooming in on it, because what's this part right here again? This is called the adrenal medulla. Okay, so what I'm doing is I'm zooming in onto the adrenal medulla and I'm looking at specifically one neuron, because you know that this actual adrenal medulla is made up of neural tissue. Specifically, they call these cells here, these neurons, they call them chromaffin cells. Chromaffin cells. Okay, now these chromaffin cells are basically going to be cell bodies of the postganglionic motor neurons of the sympathetic nervous system. Let me say that one more time. These chromaffin cells are going to be the postganglionic motor neurons of the sympathetic nervous system. And I'll explain what I mean by that. All right, so in order for us to do this, we have to start here in the actual cross-section of the spinal cord. In the cross-section of the spinal cord are about from thoracic 1 to L2 is your sympathetic component or portion of the spinal cord. Okay, from T1 to L2. They call this the thoracolumbar outflow or output. Okay? This right here is where your sympathetic nervous system is actually going to be coming out of the spinal cord. Now, the only time the adrenal medulla is really stimulated is during short-term stress. Okay, so stressful situations. In other words, fight or flight. Okay, so what is the primary stimulus of this? The primary stimulus of the sympathetic nervous system is when there is usually a short-term, or another term we could use here is acute stress. Okay, or anything that's going to try to trigger our sympathetic nervous system. So if you like to think about it like this, fight or flight, right? So for example, if I were to give you an example in this, let's say that I'm getting chased by an orangutan, okay? Little vicious little suckers, okay? That sucker's chasing after me, okay? I'm going to want to activate my sympathetic nervous system so that I can do a whole bunch of different types of physiological functions so that I can run away from that little guy. Okay? How am I doing that? Well, one way that our sympathetic nervous system is activated is through our hypothalamus. You know, our hypothalamus is actually a very strong regulator. Okay, because he has a sympathetic and a parasympathetic component. So he actually can send down fibers. He can actually send descending fibers down to the spinal cord. Where? In T1 to L2. So let's say that there's some type of short-term acute stress or some type of fight or flight situation, right? Where you need to be able to run away from that little orangutan. And this hypothalamus sends down these presynaptic potentials all the way down to about T1 to L2, which is the thoracolumbar outflow, right? Specifically right in this area. You see this area? I'm going to kind of like dot around it. You see that area right there? This component, that area, is specifically the lateral gray horn of the spinal cord. Okay. And in the lateral gray horn of the spinal cord, you see these little orange-like structures there? These little orange-like structures are the cell bodies of the preganglionic motor neurons. Okay? Again, one more time. These orange structures right there are the cell bodies of the preganglionic motor neurons of the sympathetic nervous system. So what happens is usually, if I were to follow this thing out here, usually what happens is, let's say that I have the axons coming out here, all these axons. Okay, all the axons are coming out and they're moving through, you know, this actual ventral ramus, and then eventually they go into a chain ganglia, right? So they'll go into a sympathetic chain ganglia. However, these ones are different. They don't go to a chain ganglia. They actually move right through the chain ganglia and they come to the adrenal medulla. And in the adrenal medulla, guess what I called these again? The chromaffin cells were specifically, they were the cell bodies of the postganglionic motor neurons of the sympathetic nervous system. Now again, one more time, why am I explaining this? Because normally, for the sympathetic nervous system, the preganglionic motor neurons are very short and they have to go to a chain ganglia, right? And then they'll get taken to their target organ, because then their postganglionic motor neurons are long. This time it's opposite for the adrenal medulla. It's an exception. Where the preganglionic motor neurons are very long, they pass through the ganglia, the chain ganglia, and they go out to the cell bodies which are inside of the actual organ. So as an example of what's called an intramural ganglion. Okay, so again, what would this be an example of? This is an example of one of the exceptions called an intramural ganglion. One of the exceptions of the sympathetic nervous system. Okay, now that we've cleared that up, now let's go ahead and get into all these mechanisms. So, what's going to happen? What kind of fibers are these purple fibers? These purple fibers are actually called cholinergic. They're cholinergic, meaning that they release acetylcholine. So they're going to start releasing acetylcholine, and acetylcholine is going to start binding onto this receptor here in this chromaffin cell, which could be on the cell body or it could be on the dendrites. But once it binds onto the cell, certain ions start flowing in, right? So you're going to have cations like sodium flowing in here. And when it flows in, what happens? It could activate specific action potentials which could stimulate certain processes within the cell. Look what happens as a result of these sodium ions coming in. And what kind of receptor would this be? This receptor here is actually called a nicotinic receptor. Now, once there's this action potential and it places a charge on the membrane, it might activate specific enzymes. So certain enzymes might get activated by this depolarization. And then look what happens. It might activate specific pathways that help to trigger a specific biochemical pathway. Because you know we have to make specific chemicals in this area. Look what happens. Okay, so who's going to be the first guy that we start off with up here? Okay? It's going to be tyrosine. So tyrosine is the guy that we start with. He's an amino acid. He's the building block for these processes. But then we can take these tyrosines and we can lead to this tyrosine. We can actually cause it to be converted into L-DOPA. Okay? And then L-DOPA is converted into dopamine. And then dopamine is converted into norepinephrine. And then norepinephrine is converted into epinephrine. So you know what actually this guy's secreting? It's secreting a lot of different, secreting two main chemicals out here. Two main chemicals from these synaptic vesicles. One is going to be epinephrine, okay? The other one's going to be norepinephrine. But out of these two that are being secreted or released out of the synapse into the bloodstream, 80% of it is going to be epinephrine. And 20% of it is going to be norepinephrine. All right, so you know in order for this pathway to occur, there's multiple enzymes involved in each of these steps. I'm just going to mention them for you. Again, you could actually have tyrosine to L-DOPA. This could be through what's called the tyrosine hydroxylase enzyme. I'm just going to abbreviate it for you. L-DOPA to dopamine. This is actually dependent upon DOPA D-carboxylase, so it stimulates this step. Dopamine to norepinephrine is actually through dopamine beta hydroxylase. And then this norepinephrine, this one's a heck of a name, it's called phenylethanolamine-N-methyltransferase. And it stimulates this step. Okay? So all of these enzymes are helping this whole pathway to occur. Whenever this is providing a stimulus, this whole pathway can occur and actually enhance this process here. Now normally, epinephrine and norepinephrine, they can actually be pre-synthesized and form here in these vesicles. And technically what can happen is whenever there is this sodium rushing in, it helps to be able to reach threshold for this axon. And then what happens is action potentials are actually going to be produced down this axon. Once it's produced down this axon, these action potentials, it gets down to this actual terminal bulb. And what happens is when it gets down to this terminal bulb, you know down in the terminal axon bulb here, there's actually calcium channels. And once you hit about positive 30 millivolts, these calcium channels open and calcium starts flowing in here. And then what does calcium do? It acts as the bridge between these little, called synaptoproteins, synaptobrevin and syntaxin. it actually causes these vesicles with the cell membrane to merge. And whenever it merges, guess what happens? We release out into the synapse, which is actually going to go into the bloodstream. We're going to release out the norepinephrine and epinephrine. These are the two chemicals that we're releasing into the bloodstream. So again, let's do a quick recap. The cholinergic neurons are going to be the neurons specifically from the preganglionic, sympathetic. So these are preganglionic, sympathetic motor neurons. They're releasing acetylcholine onto these chromaffin cells, which are in the adrenal medulla, which are acting like the sympathetic postganglionic motor neurons, right? So this is an example of an intramural ganglion. Then, so once this acetylcholine binds onto this receptor, this nicotinic receptor, it allows for sodium ions to flow in, right? Triggers like it's called a graded potential. Once it causes that graded potential, it brings the resting membrane potential closer to threshold, right? Once it hits threshold, here in the axon hillock, it triggers these action potentials that start moving down the axon. And then when it moves down the axon, what happens? Calcium ions flow into the axon terminal bulb and lead to the exocytosis of norepinephrine and epinephrine. Now, this whole pathway can actually be enhanced based upon certain stimuli, but usually epinephrine and norepinephrine are preformed. They're already synthesized and they're sitting down here in these vesicles waiting for these action potentials to occur. And again, if you want to remember this, it's tyrosine to L-DOPA via the tyrosine hydroxylase, L-DOPA to dopamine via the DOPA decarboxylase, dopamine to norepinephrine via the dopamine beta hydroxylase, and then norepinephrine to epinephrine via the phenylethanolamine-N-methyltransferase. Out of most of these enzymes, this is probably the more important one because he's helping to convert norepinephrine to epinephrine. So in these cells here, more of this enzyme is expressed so that you can have more epinephrine via 80% versus 20% norepinephrine. Okay, so now we've done that. Okay, how is this epinephrine and this norepinephrine that we release going to help us to run away from this orangutan? All right, let's see. You know, our actual muscles and our brain and a lot of our body actually depends upon glucose. So usually when we're in a certain type of stressful situation, our sympathetic nervous system wants to get as much nutrients into the bloodstream as it possibly can. So look what it does here. Let's say we follow this epinephrine onto this liver. And it comes onto the liver, and what it does is it stimulates a specific adrenergic receptor here. Once it stimulates this adrenergic receptor, look what it does. It activates specific G protein pathway. You already know this, G stimulatory. And then normally it's bound to GDP, which keeps it off. If it's bound to GTP, it turns it on. Then what happens? It helps to go over and activate a specific type of effector enzyme. This effector enzyme, what's actually embedded into the cell membrane, is going to do, what's its name? His name is adenylate cyclase. He converts ATP into cyclic AMP, and then cyclic AMP actually is going to activate protein kinase A. How is this significant? Well, let's see. You know there's a big storage molecule inside of our liver? About 300 grams of it is actually stored within the liver. Look at this guy here. I'm going to give you, it's a string of glucose molecules, a polymer of glucose molecules. If you guys have already watched the zona fasciculata video, you already know what's going to happen here. But this is called glycogen, right? This is called glycogen. So this molecule here is a polymer of glucose, and he is called glycogen. Now what happens is there's certain types of enzymes that are involved in this process, particularly glycogen phosphorylase and debranching enzymes. But look what it does. I can start cutting this glycogen and converting it into a whole bunch of individual monomers. What are these individual monomers here called? These individual monomers here are called glucose. Oh, I love it. So what happened? Glycogen went to glucose. What is this process called whenever you go from glycogen to glucose? This process here in a whole is called glycogenolysis, right? And if you guys remember from the zona fasciculata video, who was helping to be able to increase the sensitivity of those receptors? Cortisol, right? So if you remember, cortisol was actually helping to, well, if you remember, cortisol was that little purple molecule. Here's our cortisol. Just a tiny little correlation, it helped to increase the sensitivity of these receptors, because you know cortisol on its own actually does the reverse pathway, glycogenesis. Okay? Now, what else can this liver do? This beautiful liver. Look what else it can do. It can take amino acids. So let's say that we have amino acids here. Okay, it can take amino acids. It can take glycerol, and I'll show you where that glycerol comes from. It can take lactic acid, and it can take odd chain fatty acids. And look what it can do to it. It can take all of these things and convert all of these things into glucose. That's wild, right, guys? So what's the overall result of these guys? They get converted into glucose. So you're going to make more glucose. So what is this right here called? Again, these are odd chain fatty acids, glycerol, amino acids, lactic acid. They can get converted into glucose. What is this process called as a whole? It's called gluconeogenesis. So what is it called? Gluconeogenesis. What's the overall result? What are we putting into the blood? What's going to get put into the blood? So let's say that there's a circulation here, right? So let's say I actually draw a small little vessel here. Here's a small vessel. And this glucose gets put into the bloodstream. What happens to the glucose levels within the blood? It increases. So we have increasing blood glucose levels. What is that called when you have increasing blood glucose levels? It's called hyperglycemia. Why is that relevant to the sympathetic nervous system? Because you know your skeletal muscles can use that glucose to start helping to contract and run away from that crazy little orangutan, right? That's one reason. Okay, so that's one pathway so far. Let's see what it does on our fat tissue, okay? It comes over here and acts on the fat tissue. So let's show this, because again, norepinephrine is also going in this pathway. But again, more of it is actually going to be epinephrine. So then epinephrine comes over here, and it binds onto this G protein coupled receptor. And you guys already know this. We have already talked about this. What enzyme does he activate? You guys remember that little scissors? He activates a specific cutters. And these cutting enzymes help to be able to cut up the triglycerides. What were these enzymes called? Do you guys remember? They were called hormone-sensitive lipase. So this is called hormone-sensitive lipase, hormone-sensitive lipase, hormone-sensitive lipase. He stimulates these enzymes. If these enzymes are stimulated, what are they going to start doing? They're going to start cutting up and going chop suey on these triglycerides. And what is it going to break out? What's going to come out of it? Out of this is actually going to come what? Glycerol. So glycerol is actually going to be one component. This is actually going to be that glycerol, that head of the triglyceride, right? What was the other component that's going to be coming out of this? Fatty acids. And what were the fatty acids represented as? They were represented as these fatty acid tails, right? They're represented as these little tails. Could be like palmitoyl-CoA, right? Or palmitoleic acid, which can get converted into palmitoyl-CoA in your liver. But this glycerol, where was it going? You can utilize it there. So this glycerol can go to the liver and it can be utilized. Look what happens to this glycerol. He can actually be utilized in this pathway of gluconeogenesis. Beautiful. All right, what about these fatty acids? These fatty acids could go to the muscle, and they could actually come to the muscle, and if they're utilized by the muscle, what's the muscle going to use these fatty acids for? They're going to use it to break it down, right? Beta oxidation. And then what's the overall result of that? What's the muscles going to produce as a result? A lot of ATP. What is ATP important for within the muscles? Contraction, to allow for that cross bridge, right? So it helps with contraction of the muscle. Why? Because I got to start, cranking out I got to start running. Or I might have to, fight the orangutan, to be able to protect myself, right? So it's activating specific types of fatty acid oxidation to increase the ATP so that I can have to fight off that little crazy orangutan. Okay? So we've increased our glucose levels, we've caused fatty acid to be utilized by the muscles, and we've used glycerol and gluconeogenesis. What else can it do? Well, our heart's responsible for being able to pump blood, right? And we need our blood pressure to go high. You ever notice when you're really, really stressed or you have a fight or flight attack, what happens to your heart? It's pumping like crazy, right? Why? Because epinephrine and norepinephrine are acting at these guys too. So in the heart you have these beta adrenergic receptors, so like this one right there and this one right there. So these beta adrenergic receptors, let's say it's on the SA node. If it's on the SA node, what can epinephrine or norepinephrine do? They can bind onto this receptor. and they stimulate this beta-adrenergic receptor. By how? Increasing the calcium influx, increasing the action potentials, which does what to the heart rate? Increases the heart rate. So what's the overall result here? It's going to increase your heart rate, which does what to your blood pressure? Increases your blood pressure. It's going to activate these beta-adrenergic receptors on the contractile cardiac muscle cells and cause an increase in contractility. What happens if you increase contractility? You increase stroke volume. If you increase stroke volume, you increase cardiac output. If you increase cardiac output, you increase blood pressure. Okay, so what is the overall result of these two things? An increase in blood pressure. Let's actually show that. So an increase in this guy and an increase in this guy all results in an increase in blood pressure. Why do we want to increase our blood pressure? Because by increasing our blood pressure, we help to be able to get as much of these nutrients to the vital tissues as quickly as possible. That's one reason. But you know it also can act on specific types of adrenergic receptors, alpha adrenergic receptors, like certain blood vessels in our body. By doing that, if it acts here at these adrenergic receptors, let's say that this is an alpha-1 adrenergic receptor, what can it do to these actual alpha-1 adrenergic receptors? Cause vasoconstriction. If you cause vasoconstriction, what happens if you have vasoconstriction of the blood vessel? Vasoconstriction. What happens to the lumen diameter? It decreases. If the lumen diameter decreases, what happens to the resistance? It goes up. If the resistance goes up, what happens to your BP? Your BP goes up, your blood pressure, right? And that should make sense. Why? Because we want to get these nutrients, that glucose, that fatty acids, those glycerols, to the actual skeletal muscles or other vital organs like the heart muscle so that it can provide those different types of increased activity so we can run away or fight off that orangutan. Okay? Okay, what else have you ever noticed? Do you ever notice that, your GI tract, if you're actually having to run away from an orangutan, and let's say that you have a piece of pizza that you just ate, right? So you got a double pepperoni pizza, and you just ate it. So let's say in here is your stomach, and there's this like double pepperoni pizza. Love pizza, so good. But you have double pepperoni pizza with the cheese stuffed crust. Oh, that's the best. Okay, but you're digesting this pizza. So are you going to be caring when you're running away from an orangutan about digesting the pizza? No, you don't give a crap about that. You'll digest the pizza later. What's going to happen to the blood vessels going to the actual GI tract or the GI tract activity? It's going to decrease. So you're going to divert blood that's coming to the GI tract away from it so that it can go to the skeletal muscles or go to the lungs or go to the heart muscle or to the brain. Okay? Same thing with the kidneys and the skin. Even in your kidneys, you're going to want to constrict the blood vessels that are going to the kidneys because you'll worry about peeing later. You don't want to pee on the orangutan. You want to be able to hold that pee for later. So you're going to want to constrict the blood vessels that are going to the kidney to prevent that activity. And you're going to want to constrict the blood vessels to your skin because you're not really caring about the skin activity at that point in time. Your concern is getting as much blood and nutrients to the skeletal muscles and your heart muscle and your lungs and your brain so that you can avoid by fight or flight of that orangutan. Okay? So that's the goal there. Now, that being said, what is it going to do to your respiratory system? Okay, you ever notice that when you get really have a fight or flight situation, what happens? You start breathing really heavy, right? So it's going to want to increase the actual amount of air or oxygen coming into the lungs and increase the amount of CO2 being exhaled. So if you actually know here, you have smooth muscle surrounding your bronchi. You have some smooth muscle there. The sympathetic nervous system actually comes over here. Let's say that you have the epinephrine or the norepinephrine. It's going to come over here and it's going to act on that smooth muscle. When it acts on the smooth muscle within inside of the bronchioles, it's going to cause those bronchioles to do what? It's going to actually dilate the bronchioles. If you dilate the bronchioles, what's going to happen to the amount of air that can flow in and out? It's going to increase. So you're going to have an increased airflow going in and out. So there's going to be an increase in respiration. So what will happen here? What's the overall result with the lungs? There'll be an increase in respiration. By how? What's the mechanism by which it does it? It dilates the bronchioles. Okay? So that would be working by some type of usually alpha-2 adrenergic receptor, right? All right, Ninja Nerds. So throughout this video, we covered a lot of information about the adrenal medulla and the pathway by which it's being synthesized for the norepinephrine, epinephrine. We talked about its release. We talked about its stimulus via the sympathetic part of the actual spinal cord from T1 to L2. We talked about its effects on the target organs and all of those amazing and beautiful effects it does. And you guys can imagine that any kind of condition in which there could be like a tumor in this area and you're producing excessive amounts of epinephrine and norepinephrine could be potentially disastrous on specifically your blood pressure and your glucose levels and your respiration. If you're interested enough, this is called pheochromocytoma. Okay, it's whenever you have a tumor within the adrenal medulla. It's usually a neuroblastoma, and it causes excessive amounts of epinephrine and norepinephrine release, and it's very, very dangerous. So I really hope all of this made sense, guys. I hope you guys really enjoyed it. In the next video, we're going to basically cover a lot about all of the hyper and hyposecretions. I hope you guys enjoyed it. Until next time, Ninja Nerds.